So the second speaker is Dr. Hyman Hartman. Dr. Hartman is also an alumnus of McGill University, like Professor Zostek, following which he went on to pursue his PhD in biochemistry at Columbia. He currently works at MIT's Department of Biology and has groups in both the University of Kentucky as well as in McGill University. Dr. Hartman began his early studies on the origins of life, where he published pioneering papers as early as 1974 on the evolution of the genetic code and the evolution of metabolism and photosynthesis. These papers were based on the clay theory for the origin of life. His work on ribosomal proteins, as well as on amino acid tRNA synthetases, have allowed reconstruction of the origin and evolution of the translational apparatus. He currently studies the de novo synthesis of clay as catalyzed by amino acids and decarboxylic acids, all of which we'll be discussing later on today. Dr. Hartman has co-authored a number of books and was co-editor of the book, The Search for the Universal Ancestors, published by NASA. Could we all just put our hands together to invite Dr. Hartman? Thank you.
As someone said, why am I here? I don't know, but I was invited by Sydney, and that is such an honor that I've designed this whole talk for him. And I hope you will also benefit from that. So what I want to talk about today is basically about the genetic code, metabolism, and clay, but given the time limit, it's mostly going to be on the genetic code and metabolism. And what I want to give you is a complete theory for the origin of life that began in 1971. Now, the actors you've already seen are, of course, the nucleotides. Oops, sorry. No. Why am I not going backwards? Oh, here. Are the nucleotides of C, A, U, and G. G and A are, of course, purine nucleotides. C and U are pyrimidine nucleotides. So those are actors number one. Actors number two in the genetic code are the amino acids. 20 amino acids. And the problem, of course, is how do you code for 20 amino acids using 4 bases? Because after all, the genetic code is a dictionary of how a sequence of bases or nucleotides code for amino acids. Can't use one base, can't use one nucleotide. 2 will give you 16, and 3 will give you 64. Now, I want you to notice a few things about the amino acids. They have properties that deal with the solubility of the side chains in water. So they're hydrophobic, they hate water. These are hydrophilic, and they love water, and these are in between. Now, I want to bring your attention to these 2 positively charged amino acids, lysine and arginine, and count the number of carbon atoms up to the NH group. Arginine has 3. Lysine has 4. There is a precursor of arginine, which is called ornithine. And the question is, why was there ornithine and lysine? That's a problem I want to talk about later. The other 2 things I want to say is these 2, tryptophan I would consider hydrophobic, and tyrosine is closely related to phenylalanine. Other than that, we have this hydrophobic, hydrophilic, neutral. So this is the genetic code that was originally written by Crick in 1966. So the way you read it is, this is the first letter in the codon, this is the second letter in the codon, and this is the third letter in the codon, right? 3 bases that will code for an amino acid. And the code is written as follows. U, which has the middle base, is all hydrophobic. Phenylalanine, leucine, isoleucine, methionine, valine. Those are the hydrophobics I showed you. If we go to A, it's glutamic aspartate, lysine, asparagine, glytamine, histidine, and tyrosine. These are hydrophilic. They love water. This one is the neutral one, alanine, threonine, proline, and serine. These are the neutral amino acids. Now I want you to look at G as the middle codon. It's got cysteine, tryptophan, arginine, serine, arginine, and glycine. There is no rule on that thing. So one night in 1971, it was New Year's Eve on 1971, I rewrote the code as follows. First base was G and GGCA. So instead of U and C, it's G and then C and then A and then U. That's all. Nothing has changed. I've just reorganized the columns and the things, the horizontal. Now, at that moment, I had my epiphany. That's where the origin of life became transparent to me, and I hope to you. Because looking at it this way, here's what I saw. Whoops. I fell asleep at that time. That here's how the system grew. It grew like an onion. You start off with GG coding for glycine. Oops. I'm sorry about this. I'm always mixing up. GG codes for glycine. You bring in C into the coding system, and you get alanine, proline, and arginine. Then you bring in A, glutamine, histidine, and arginine, serine. You're bringing in the hydrophilics. And then U comes in and brings in the hydrophobics. So there, right there in that simple moment of just reorganizing the code was going to determine the rest of my miserable life. Because that pattern showed me something that no one had seen before in the code. That the history of the code was within the code itself. You didn't have to go into fancy chemistry. Right there was the history of the code. And so this is what I wrote in 1975. An evolutionary scheme is postulated in which the bases enter the genetic code in a definite temporal sequence, and the correlated amino acids are assigned definite functions in the evolving system. The scheme requires a singlet code for glycine, a doublet code for glycine, alanine, arginine, and proline. The doublet code evolves into a triplet code, and the polymerization of nucleotides is thought to have been by block polymerization rather than by template mechanism. The proteins formed at the first were simple structural peptides. No direct nucleotide amino acid stereochemical interaction was required. Rather, an adapter-type indirect mechanism is thought to have been functioning since the origin. Now, the problem I told you about, lysine and arginine, presented an interesting question because arginine, of the 4 amino acids in the GC code, glycine, alanine, and proline are relatively simple amino acids, and they're easy to make. What is hard is arginine. And the question is arginine a later evolved positively charged amino acid? And so was there something smaller than ornithine, which is the precursor of arginine? And there are a number of possibilities. Whoops. There is the 2,4-diaminobutyric acid, which has 2 carbons instead of 3. And then there's my favorite, the diaminopropionic acid, which has only 1 carbon. It's basically alanine with an NH2 group, exactly like serine is an OH group and cysteine has an SH group. And now if you put in diaper, which is a nice thing to use, suddenly the G column makes enormous sense. Because glycine, diaper, and diaper sharing a codon with serine is essentially NH2 and an OH, and cysteine also belongs in that group. And suddenly all the columns now make sense. So what were the lessons that I learned from this? The point is that this thing grows like an onion, and you start simple, you keep adding onto the thing, and it grows like an onion. That means in the complex systems that you find in biology, and which are quasi-universal. So all the complex alternatives have been cut off. This logic will work in every case. So it was quite clear, this was in 1971, the next year it was clear that this logic should be applied to metabolism. And so before I go into that, if something is growing like an onion, it had to evolve. And the question is, what was the replicating, mutating material? Well, this was presented to me in reading Journal of Theoretical Biology by Karen Smith. And this was written in 1965. And he said, according to the specific theory as proposed, the primitive genographs were patterns of substitution in colloidal clay particles, crystallites. The theoretical information density in such a crystalline is comparable to that in DNA. Evolution proceeded through selective elaboration of substitutional genographs, that is, survival for the clay crystallites that held them, at first through genetically controlled absorption of a spectrum of organic molecules within a complex, dynamic, primitive environment, a gradual takeover of the control machinery by organic molecules, a genetic metamorphosis, would evolve from a simple, well, hold on a second, but is considered to have occurred. Graham Karen Smith just died last August, and he and I wrote a book together, edited a book together on clay minerals and the origin of life. So this, what I read then, did blossom forth in a collaboration with him. All right, so metabolism. The second great universal is metabolism. And so what I proposed at that time was that an autotrophic, that means that you got all the metabolic stuff like we do today, from CO2, nitrogen, water, sulfur, phosphate, etc., is described, which requires clays, transition state metals, disulfide and dithiols, UV, and cyanide ion. A general scheme is proposed involving the fixation of CO2 and nitrogen for the evolution of an intermediary metabolism based on the evolution of a complex system from a simple one. The basic conclusion is that metabolism could have evolved from a simple environment rather than a complex environment. No soup. This is now starting directly from CO2 and nitrogen and is going to build up metabolism. And when you look at this is from the paper in 1975. When you look at metabolism, what is absolutely clear in anabolism, that means when you're making molecules from the citric acid cycle, it is that the citric acid cycle is the center from which you now build all your amino acids, all your nucleotides, etc. And that there is a metabolic metric which tells you how many steps does it take to make an amino acid from the citric acid cycle. And what is interesting, it takes things like glutamic and aspartic acid, very, very small steps. Glycine is made easily. Proline is made easily. Arginine is more difficult. That's why I proposed that there were precursors to the growth of iron. So this is it. I always keep pushing on. If we look at intermediate metabolism, in particular anabolism, we are immediately struck by the central play by the citric acid cycle. In the anabolic things of the citric acid cycle, we apply a method of Bernal, which I'll describe to you. It can be conjectured that the citric acid cycle came first and was followed by the amino acids, lipids, nucleotides, and carbohydrates. From a more detailed examination of metabolism, it is evident that some amino acids require fewer steps in their biosynthesis from the citric acid cycle. Glutamic acid, glutamine, proline, arginine are closely linked metabolically to alpha-ketoglutarate, aspartate, etc., are linked to oxaloacetate, alanine, etc., are linked to pyruvate. Glycine is also made from glyoxylate. There is a puzzle with respect to lysine, for it is easier to make ornithine from the citric acid cycle. One might speculate that ornithine came into the biosphere first, later to be supplanted by arginine and lysine, and then I took it a step further and took it down to diaper. Yeah, the amino acid histidine is closely associated with the biosynthesis of purines. Okay, so what did Bernal say? And the idea is exactly what I just said to you. It is, he said, the method describes a complex system evolved from a simpler system. The pattern is one of stages of increasing inner complexity followed by one another in order of time, one including itself structure and processes evolved at the lower levels. The system thus grows more complex like an onion, and it must be possible to strip back the layers of the onion to include the simpler stages. So to solve the origin of life, strip back the genetic code, strip back metabolism, and then hope that clays will evolve it for you. Now, the big problem, this is the citric acid cycle, just for your benefit. It's a set of dicarboxylic and tricarboxylic acids. These are the amino acids coming off the citric acid cycle. These are the ones that take a lot of steps from the citric acid cycle. Arginine, lysine, methionine, isoleucine, leucine, tyrosine, phenylalanine, tryptophan, and histidine. These are the ones that are longer from the citric acid cycle. These are the ones that are in the outer edge of the genetic code. So the genetic code and the amino acids in the genetic code are completely tied into the metabolic metric from the citric acid cycle. Now, the big problem in this system, in driving metabolism, you need an energy source. Where does the energy come from that drives metabolism? Of course, it's photosynthesis. And now one would like to do is now recover the evolution of photosynthesis. Now, this is a harder problem than metabolism or the genetic code, because This is not universal. There's only special organisms like cyanobacteria and purple photosynthetic bacteria and green sulfur bacteria. So the question now is to try and reconstruct how it goes from an electron donor to CO2 and nitrogen. That is the problem. And to make a long story short, that took me approximately 1992. That is, from 1971 to 1992, trying to reconstruct the evolution of the photosynthetic system, that is a harder problem. And I'll just tell you what I found, and you can read this paper. It's brilliantly written. The origin of photosynthesis is postulated to have involved carbon dioxide and self-replicating iron-rich clays. The latter evolution of photosynthesis is considered to have undergone 4 distinct phases. 1, the photoreduction of CO2 by ferrous iron to form oxalate and formate. 2, the entry of sulfur into the evolving clay system, which led to the formation of acetyl thioesters. The polymerization of the acetyl thioesters led to things like the formation of quinones and the formation of FeS2 and FeS4, ferredoxins, cores, appeared in this phase. The ability to fix nitrogen, which is one of the toughest problems still to solve of how nitrogenase came into existence, characterized the 3rd phase. This led to the formation of pyrroles, flavins, nicotinamide, psychobilins, porphyrins, and chlorophyll. 4, finally, phosphate entered the evolving system. Phosphate came in last. The chromophores evolved from ferrous iron through the quinones, carotenoids, phycobilins, to chlorophyll. The evolution of chromophores implies that photosynthesis began in the UV and evolved through the blue, yellow, orange into the red. The electron transport chain evolved from ferrous iron through FeS2, Fe4S4, and cores to the hemes. So therefore, what photosynthesis tells us is that the elements came into the biosphere not all together, but CHO came in first, then sulfur, then nitrogen and ammonia, and then phosphate. And so far, I'm just making the claims that I had up to that time. And at that particular time, this is the paper I wrote in 1998 in Photosynthesis and the Origin of Life. Forget about the top part, it's just a repetition of what I just said. So if there was no soup and life began as a photoautotropic iron-rich clay on Earth, then when we sample the surface of Mars for fossils of early life, we should look not only for amino acids and other biochemicals, but also for the ancient fossil minerals such as iron-rich clays and magnetite. And you should know now that the biggest finding on Mars is that the first 500 million years on Mars are called the phyllosilicate era, and they are dominated by iron-rich clays. So this has cosmological implications. All right, the origin and the evolution of the genetic code. And now I'm going to take you to 2016, which was a meeting that was held in Cold Spring Harbor on precisely this problem. And what has happened to molecular biology since I had this enormous vision in 1971? I just want to stop here for a point. If you're given a gift like I was given on the origin and evolution of the genetic code, you then may have to spend the rest of your life proving it to yourself that you were right. And that means you have to look for data. And that's what happened at this meeting in Cold Spring Harbor. So in 1968, wrote the following thing. It is almost impossible to discuss the origin of the code without discussing the origin of the actual biochemical mechanisms of protein synthesis. This is very difficult for two reasons. It is complex, and many of its details are not yet understood. Now that's where this thing stood until 2001, when they had a big meeting at Cold Spring Harbor on the crystallization of the ribosome. So this was the meeting. The title of the meeting is The Evolution of the Translational Apparatus and Its Implications for the Origin of the Genetic Code. Now just let me remind you what's involved in protein synthesis. In protein synthesis, it's at the ribosome that the messenger RNA is translated into the protein. That's the head that's reading the tape and converting one tape into another tape. The interesting point, as was pointed out earlier, that there is a peptide transfer center where these transfer RNAs are activated in one case on the A site with an amino acid and with a polymer of amino acids on the other site, the P site, and then there's a transfer of the polymer to the A site. This thing goes out, and now you have one more amino acid, and then a new tRNA with an amino acid comes in. I'll talk to you about the nucleotide transferases later. It has to do with CCA enzyme and poly-A enzymes. This is the tRNA pool, and this is the enzyme, the aminoacyl tRNA synthetases, that put the right amino acid onto the tRNA, and so that the anticodon, which is here at the other end of the tRNA, can interact with the message, the three nucleotides on the messenger RNA. It's an interesting story. The transfer RNA is the system where the amino acids are on one end, the anticodon is on the other end. It goes into the ribosome. It reads the message, the three bases on that thing. It then passes, the peptide then passes to the A and grows by one amino acid. It's a beautiful piece of machinery, and it's huge. That meant it had to evolve. It could not have begun in this huge, complex form, and the question is, can one get at the evolution of the ribosome? So I'm going to start off, and here's the rest of the talk. I'm going to start off with the ribosome. I'm going to go back to the aminoacyl tRNA synthetase and its evolution, and then to the evolution of the tRNA based on these nucleotide transferases, the CCA enzyme that adds CCA to the tRNA. Let's go. The ribosome. Here is the ribosome in its glory. There is a small ribosome and a small subunit and a large subunit of the ribosome. They're big. There's approximately, for E. coli, 3,000 nucleotides in the RNA of the large ribosome, 1,500 nucleotides in the small ribosome. Here it looks like where the proteins are in blue. And you'll see that this is the large subunit looking at it from the inside. And you can see that there are proteins that are sending peptides into the RNA. And we'll come back to those later. Now, it was immediately seen that the ribosome did grow like an onion, and it grew from the PTC site outwards. And it grew by addition of other pieces of RNA. This is the peeling the onion. the onion comes up a lot in this business. And the ribosomes are ancient molecular fossils. And now the problem is, how do you read the onion-like growth of the ribosome? So Yonath looked at this and said, how did the PTC site come into existence in the first place? And her idea was, you start off with two RNAs that are just small RNAs. They dimerize and ligate. They then form the PTC site, the peptidyltransferase site. They're at the center, and then it grows out from there. Now, my friend Petrov and Lauren Williams have spent the rest of their time seeing how this thing grows from the PTC. And it's small RNAs constantly being added onto. It's definitely growing like an onion. It starts off with the PTC, and RNAs are now ligated onto this thing, and that is how this thing grows. Now, answer number one. The ribosome is growing like an onion from a center, from the PTC center. And its pieces are being added onto it. So what is true about the ribosomal proteins? So everyone in 2001 went for the RNA, the PTC, and the RNA of the ribosome. And Temple and these graduate students and Vittel decided to look at the proteins. What was the nature of the proteins in the ribosome? Now, having crystallized the ribosome, they had the structure of every single protein down to the 1.8 angstrom level, 2 or 3 angstrom level. So the proteins are of great interest. Now, the proteins are as follows. At this moment, I just wanted to say something, because I did say something about eukaryotic cells. So I just want to step aside for a moment. The question is, where does the eukaryotic cell come from? Where does it come from? It has a nucleus, and the bacteria-like system, the prokaryotes, do not. And there is bacteria, and there's archaea, and there's the nucleus. And there is the eukaryotic cell. So most people think that the eukaryotic cell is some fusion of archaea and bacteria. That is not my picture. My picture is that the archaea were endosymbionts that led to the nucleus. The mitochondria and the chloroplasts came from the bacteria. When you subtract out all that stuff, you're left with an RNA-based organism. So this may be the organism that Shostak is looking for. But that's another story. The major point here is to look at the ribosomal proteins. The distinction between archaea, bacteria, and eukaryotes all came from the small subunit RNA by Carl Woese, which was a magnificent achievement. Well, here is what is true. The eukaryotic cell has 78 ribosomal proteins, 32 in the large subunit, 32 in the small, 46 in the large. Archaea has 68, 28 in the small, 40 in the large. It turns out 68 of the archaea are homologous to 68 of the eukaryotic cell. From the ribosomal point of view, the ribosomal proteins is that the eukaryotic ribosome is an archaeal ribosome with 10 extra proteins. Now, the big difference comes in the bacteria. The bacteria have 57 ribosomal proteins, 23 in the small, 34 in the large. Now, in order to do phylogeny on these ribosomal proteins, you've got to have universal ones so that you can align them and you can look for how they align. And it turns out that there are 34 universal that are in bacteria, archaea, and eukaryotes, 15 in the small and 19 in the large. And we did then take those proteins and align them and looked at their alignment. And the alignment immediately brought a huge surprise, because in the same protein, there were blocks that were archaeal, blocks which were bacterial, and common blocks. That meant there was a piece of that protein that goes back to a universal ancestor of the archaea, a piece of that protein which goes back to a universal ancestor of the bacteria, and then there was one that went in common. There was a deeper one, which meant that these proteins were put together in pieces. And they were put together differently in the archaea than they were in the bacteria. And so I want to spend a little bit of time talking to you about 4 proteins that are in the large subunit. These are L1, L2, and L3, and L4, and L22. Now, when ribosomal RNAs fold, Nomura showed that certain proteins go on first, and then there's a whole cascade of how the RNA folds. And the ones that are directly related to the ribosomal RNA is L24, whoops, I'm always doing the wrong, is L24 at the N terminal and L14 at the C terminal. But L1, L3, L2 definitely are coming, and L4. So L1 is one thing, L2, L3, and L4, and I'll talk somewhat about L22. These proteins are there at the earliest stages, directly in the folding of the ribosomal RNA. And a number of people have claimed, George Fox among them, that means that these proteins are also the most primitive of the large subunit proteins, because they're involved in the actual folding directly on the RNA. Now here's one of the things, these are L4, that shows you sort of the block structure I was talking to you about. These things are common block structures. These things are with the bacteria. These things are with the archaea. Here's what it looks like when they fold up. This thing is the common region. And then there is this region where it's sort of a similar structure, but the sequences of the amino acids are very, very different. There's where I told you that we have this interesting problem in the ribosomal proteins, that they're built out of pieces. And here's how they interact. Here is L2, L4, L3, and I'm also going to talk about L22. I'm not going to talk about this one because it seems to be a mimic of a tRNA. So what we did, it turns out that the amino acid composition of these extensions are totally different than the globular forms. That means that these proteins have broken up into 2 pieces. One is the extension that goes into the PTC site, and the other one is the globular form that's on the surface. And so we looked into the amino acid composition of this system, and it turns out that they are fantastically enriched in alanine, glycine, lysine, proline, and arginine. These are the amino acids that dominate this system. The other thing that you'll notice about this system is there are no alpha helices. These are loops with a lot of disorder. So our hypothesis is that the memory of the GC code is found in the peptide extensions of the universal ribosomal proteins L2, L3, and L4 of the large ribosomal subunits. These ribosomal proteins have peptide extensions that are found at the RNA center of the large ribosomal subunit, the peptidyl transfer site. The evolutionary expansion of the LSU from the PTC can be correlated with the expansion of the code, GC, GCA, GCAU, as seen in the structure and amino acid composition. What the hell? Sorry about that. As seen in the amino acid composition of the peptide extensions at the RNA of the PTC, followed by a beta hairpin extension of L22, interacting with the RNA of the tunnel, and finally with the proteins L23 and L29 and their interaction with RNA at the exit of the tunnel. We conjecture that the evolution of the secondary structure of the polypeptides began with loops with beta turns, followed by beta hairpins, followed by alpha beta proteins, and then by alpha proteins. The evolution of the secondary structure of the polypeptides correlates well with the expansion of the code and the entry of amino acids into the evolving system. That is, in the evolution of these proteins from the PTC site, as they go out to the periphery, you can see the evolution of the folds, and that the actual evolution of the folds seems to be ending up at its best with alpha helixes. All right, the summary of this meeting. Forget about what I just said, forget about everything else. There was one summary of this meeting, that in the translational apparatus, in the ribosomal proteins, in the ribosomal RNA, there was the simple idea that the translational system evolved out of a world of small peptides and small RNAs. And it bootstrapped itself by ligation. So I thought that that was something rather interesting. That is, when you look at something like the ribosome, you can reconstruct its growth. And if you look at the proteins, you can see the evolution of folds. All right, now we come to the heart of the genetic code, the amino-acyl tRNA synthetases. There are two of them, two kinds, class 1 and class 2. There are 10 amino acids in each one of them. These are dominated by the hydrophobic amino acids. There is a glutamate, glutamine, lysine thing, and then there's tyrosine tryptophan. The class 2 in this system here, serine, threonine, alanine, glycine, proline, and except for histidine, these are the amino acids that I'm going to concentrate on. Aspartate and lysine are involved here, phenylalanine, and those are the two codes. It's usually thought that the class 2 synthetases came before the class 1s because of the simplicity of the metabolic metric by which you make these amino acids. This is what the synthetases look like. And they are made out of modules. The blue modules are the ones that look at the tRNA and see the anticodon. The upper modules are made up of the editing module and the catalytic module. The catalytic module is where ATP interacts with amino acids, makes them AMP amino acid, and then it's passed on to the tRNA. There are two types of things. And so this is how it looks. Here's the editing site, there's the activation site, and there's the anticodon binding site. A side note. Now, here's an interesting fact. It was determined by the work by Jakubowski many years ago, that there was a site on the catalytic site that accepted the activated amino acid and made them thioesters. That they took ATP plus an amino acid. There was a site on there where it was something like cysteine, and you would get a thioester. So this has been something that's been lying around for quite a while. Now, this is the catalytic site of glycine, alanine, aspartate, proline, and these are the bacterial glycines. There's two glycine catalytic sites, which is still interesting and controversial. But the interesting one is how similar the glycine one is to the proline one. It's different than the alanine one, and this is different on its own. Again, we're now going to do the trick that we did with the ribosome. These things are being built out of smaller stuff. So if you now look at the active site of these amino acyl tRNA synthetases, they are built out of a number of hairpins. The actual active site is this loop here. And then when you look at those entities, the one with the active site, that loop, and we look at the sequences, etc., and the structures, we end up with this tree. And the tree has glycine and proline going together. Lysine, well, it's always... lysine and aspartate go together, and then alanine and this other glycine go together. If you look at this tree more carefully, you'll see that these are probably more primitive. These came slightly later, and these are derived. So we are going to concentrate on the glycine-proline case. So this just tells you what I just said. The amino acyl tRNA synthetases are a set of very ancient multi-domain proteins. The evolution of the catalytic domain of class II synthetases can be reconstructed from 3 peptidyl hairpins. Further evolution from this primordial catalytic core leads to a split of the class II synthetases into 2 divisions, potentially associated with the operational code, which I will come back to. The earliest form of this code is likely to have coded predominantly glycine and proline, with later alanine and lysine aspartate. There is a paradox in these synthetases. Beginning with a hairpin structure before the genetic code existed, a resolution is found in the suggestion that the primordial amino acyl synthetases formed in a transition from a thioester world to a phosphate world. This is Jakubowski in 1998, and I just want you to read the last thing. He talks about these and other days support a hypothesis that the present amino acyl tRNA synthetases originated from ancestral forms that were involved in non-coded thioester-dependent peptide synthesis, functionally similar to the present-day non-ribosomal peptide synthesis by multi-enzyme template systems. You will know that Lippmann discovered that many of the antibiotics, like gramicidin, are made by the polymerization of thioesters and a non-coded form. There is a non-coded peptide synthesizing system that is not based on messenger. And it leads to complicated things like gramicidin. So, and then it's recently been shown that these catalytic sites are all over the cell now, independently of the tRNA synthetases, and they are there to make thioesters. So now let's enter the thioester world. Does such a world exist? Can you prove such a world? Okay, this was done by Deduve. He was the first man to claim that this system is whoops, it's always the same. It is revealing that the thioesters are obligatory intermediates in several key processes in which ATP is either used or regenerated. Thioesters are involved in the synthesis of esters, including those found in complex lipids. They are participants in the synthesis of a number of other cell components, including peptides, fatty acids, sterols, terpenes, porphyrins, and others. In addition, thioesters are formed as key intermediates in several particularly ancient processes that result in the assembly of ATP. In both these instances, the thioester is closer to ATP to the process that uses or yields energy. In other words, thioesters could have actually played the role of ATP in a thioester world initially devoid of ATP. Eventually, these thioesters could have served to usher in ATP through its ability to support the formation of bonds between phosphate groups. Okay, so here's what we did. So this is a paper hot off the press. It was just published in Cell in 2017. So this is hot off the press. It is the remnants of an ancient metabolism without phosphate. How would you try to show that? So, as someone once pointed out, I think it was Shostak, how complex metabolism is. And of course, it's evident now that you've remember that the citric acid cycle is right there. And here is the metabolic chart, and it's in the KEG group. It's an enormous metabolic chart. So here's what you do. You start off with a seed of what you would think was the primitive materials that went into metabolism. CO2 and water, well, of course. HCO3 because it's soluble. H2S, of course. Nitrogen, ammonia, and formaldehyde and acetaldehyde. Put that in to the KEG system. Here, you put it in, and then you see, how does it flow? Where does it go? Without phosphate, you throw it any phosphate at all, and you get a very complex metabolic network, which has a lot of the interesting glycine. It is a fairly complex network. In fact, you can go further and say, what happens if I leave out nitrogen and ammonia? And you get a smaller network. And then you say, what happens if I leave out sulfur? And I get a smaller network. And you end up with a CHO, and then you can expand it with sulfur, and then you expand it with ammonia, and you expand it with phosphate. So what I had predicted in the evolution of photosynthesis is completely here in this metabolic chart. That means the history of metabolism is still there in the metabolic chart. Now, is this important? Well, I'll tell you yes, and I'll tell you why it's important. Because it means that these systems are still operating in our cells. And what is even more important, that they may have enormous control over the metabolism of the cell. I'll give you an experiment that I did a number of years ago with Bob Buchanan. Thyrodoxin is a small 110 amino acid protein, and all it does is open and close disulfide bonds. It turns out that that thyrodoxin is involved in the conversion of ribose to deoxyribose, some free radical mechanism. So Buchanan and I and John Gerhardt decided to try different thyrodoxins into xenopus eggs that were fertilized, that go on to 13 divisions and then go through the mid-blastular transition. Well, we did things like cyanobacteriothyrodoxin, E. coli thyrodoxin, and put it in. This thing went through 13 divisions. We opened it up, and there was no nuclei. It had gone through all this mitosis. That means there is a system in the eukaryotic cell that is under the control of disulfide and dithiol. In plants, it's very prevalent. So the real question underneath it, today, our whole idea of the control in the eukaryotic cell is kinases and phosphatases. What about disulfides and sulfhydryls? What does arsenic do? Because arsenic ties up disulfides, dithiols. So these ideas that are now being discussed about the origin of life does have, I believe, humbly, impact on medicine. Okay, so this is just a conclusion, a phosphate-free. I have another 5 minutes. Is that possible? Is that possible? All right, so this is just this shows you that the Krebs cycle could be done with thioesters. And now I want to talk about Shostak's problem of RNA. And it has to do with the origin and evolution of the tRNA. And now I call it the origin of the RNA province as opposed to the RNA world. That means RNA is not coming in at the origin of life. It's coming in into a system that was already sufficiently complicated. So the peptides that he's hoping and praying for were there all the time. That is not an RNA-free system. That's just my humble opinion. This is the tRNA. You will notice one thing about the tRNA, and that is that there are 4 invariants in the tRNA. There's the UCCA part of the arm. There's the T5CG, which is really UUCGA. And then there's the UGGU, and then there's the anticodon system. Now, you will notice that at the CCA end, the CCA is added by an enzyme called the CCA enzyme that is not coded for. It's added on by the CCA enzyme. And this is my hypothesis that the modern tRNA is a tetramer, and it's in the loop, and the evolution of RNA can be followed as follows, that it was preceded by UGCA, UGGA, UCCA, UCGA. It's further thought that it may even have gone down to these triplets. Here's the CCA enzyme. This is what it looks like. This is, I'm not going to trouble you with all that. It's made again by these small peptide systems. The idea then that such peptides existed and were ligated together and formed the CCA enzyme. This is from the Arvind Kunin paper. It turns out that that simple motif is the motif of a huge family of nucleotide transferases, the poly-A that's added on to message, etc. So the point is, here's the hypothesis. The first RNAs that were made were either trimers or tetramers. And that is the first RNA that was not coded for and was essentially synthesized by peptides. And now I want to tell you about the operational code. That has to do with the arm. I'm sorry. It has to do with the arm. And that, of course, tells you that these small RNAs, when added to amino-acyl tRNA synthetase, put on the right amino acid because there's a code in the arm. And that was shown by Rodin and Susumo, that there was an archaic GC code in the arm. The general thesis has been argued that, like other evolving systems, the code began simply and evolved to the present complexity. The very fact that anticodon-codon-like triplets in the acceptor helix turn out to be composed of predominantly GC and CG bases strongly supports this hypothesis. And this is how Uno and Rodin grew their tRNA, and this is how the tRNA, the CCA system evolved up a system that read the operational code. And this is nothing about. And now it just brings us back to this thing, that the evolution of the tRNA is the key to the evolution of the RNA world. And this is, of course, the three-dimensional structure. And with that, I will end. I have one short comment, if I could make. So the most recent spectacular finding that has been found relative to tRNA is tRNA is fragmented, makes fragments. This was the conference at Banbury. And this is the thing I would have talked about if I had time, but that would be another hour and a half. And this is just a gift to Sostak that in some basic sense, he made his most significant experiment by putting clays into his membranes. And that you can make membranes from polyglycine. And that's all. The spectacular finding that I want to tell you about is there is this thing about tRNA fragments. tRNA is being fragmented into pieces. One of those fragments is involved with obesity. If the father is obese, this fragment of the tRNA goes into the sperm, and the progeny will all be obese. This is epigenesis. So tRNA is not your simple RNA. So keep your eyes and ears open for small RNAs that are running around the eukaryotic cell. That's all. I just wanted to make that one point.